So in my talk, I will introduce two in vitro systems that can be used to assess the genotoxicity of CRISPR-Cas9 genome-edited cells. And I will start with a quick introduction to CRISPR-Cas9. So the CRISPR-Cas9 system consists of a CRISPR RNA that has a 20 base pair sequence that is homologous to the human target sequence. And then the CRISPR RNA is aligning with the tracer RNA, and it's forming a complex with the Cas9 protein. The Cas9 protein is then guided to the human target sequence, where it introduces a double-strand break. The double-strand break can be repaired by non-homologous end joining, which is error-prone. So this leads to a gene knockout. It can also be repaired by homology-directed repair if a DNA template is available, and this leads then to a gene knock-in. So this is now a super easy system which is broadly used, and therefore it's also suitable for gene therapy. However, for the applications in humans, there are some safety concerns. For example, the editing can not only happen in this human target sequence, but somewhere else in the genome. And then this is called off-target editing. And this can be dangerous because, for example, if this happens in an oncosuppressor, which is then knocked out, it could lead to malignant transformation. And it has been shown that the CRISPR RNA shows a partial could show a partial homology to the target sequence. So the off-targets are sequence dependent, which is why for off-target analysis, we need a human cell-based system. So how can we evaluate the safety of CRISPR-Cas9 genome-edited cells? There are no fixed guidelines available. However, there are a few approaches. For example, first of all, the off-targets need to be detected by in silico analysis. So these are softwares that detect off-targets based on their sequence homology to the human genome. However, these softwares are not perfect. So there is a need to follow up with human cell in vitro-based systems. And these are mostly next-generation sequencing-based methods. Here, the information about functional consequences is missing, however. And to assess these functional consequences, commonly there are in vivo tumorigenicity studies performed. And for this in vivo tumorigenicity studies, the human edited cells are injected in immunocompromised mice, where they are observed for tumor formation for at least 6 months. However, it has been shown that the engraftment rate of human tumors in NSG mice is highly variable depending on the tissue type and the aggressiveness of the tumor. And they are also time and cost consuming, and they bring discomfort to the animals. So in this case, it would be very advantageous to have an in vitro system that can detect the functional consequences of the off-targets. So how did we develop such a system? We were first looking for assays, in vitro transformation assays that were already available. And we are focusing on assays that were based on anchorage-independent growth, because this is a feature of transformed cells. And we found 2 assays, the soft agar colony forming assay and the growth and low attachment assay. And I will explain them in more detail on the next slide. But here it's important to keep in mind that these assays are only suitable for adherent cells and not for cells in suspension, because we focus on this one main feature of transformed cells, which is the anchorage-independent growth. So for the soft agar colony forming assay, the cells are seated in agarose in a single cell suspension layer. And there they don't find any attachment. So normal cells die and the transformed cells will be able to grow. And after 4 weeks, we stain the colonies, fix the cells, and solubilize the agar so that all the colonies gather in one plane, which enables image analysis. For the GILA, the cells are seated in ultra-low attachment plates where they also don't find any attachment. And again, only the transformed cells will be able to grow. And after 2 weeks, we lyse the cells and detect the ATP levels. So these are now 2 super easy systems. And then we wanted to generate a really robust assay. So we started to validate every component of the assay. For example, the right cell number or the best statistical methods. And here I will give you an overview of all the validation we did regarding genome edited cells. So we needed a suitable surrogate cell line. And in this case, it would be a human cell line because of the sequence dependency of the off-targets. And it should be an immortalized but not transformed cell line, because the cell line should not be able to grow in soft agar because we want to make it grow by editing them. And there we found the MCF DNA cells, which are human epithelia breast cells. Then we wanted to have an easy experiment design. So we generated a cell line that's already expressing Cas9. So we just have to electroporate them with the CRISPR tracer RNA complex. And then subsequently, we could determine cleavage efficiency and perform GILA and soft agar from the same cell pool. Then we needed suitable controls. And in this case, a suitable control would be a CRISPR-Cas9 mediated control. So as a positive control, we would delete genes that transform MCF DNA. So for example, oncosuppressors. And as negative control, we would delete genes that don't have an effect on the anchorage-independent growth of MCF DNA. And we determined a set of positive and negative controls. And in this talk, I will focus on PTPN12, the strongest positive control in our hands. And I will show you some data about the limit of detection and the comparison to the standard animal experiment. So as I already mentioned, PTPN12 is one of our strongest positive control. It's a known oncosuppressor, and we've shown previously that PTPN12 is inducing hyperproliferation in MCF DNA. So then we wanted to validate if it can also induce anchorage-independent growth. So we chose 3 CRISPR RNAs that have a high cleavage efficiency and verified their protein verified if they efficiently knock out the protein. And then we tested them in soft agar and GILA. So here you can see the soft agar in colony count, and here the GILA in ATP levels. And in both cases, all 3 CRISPR RNAs induced significant anchorage-independent growth. And CRISPR RNA1 was performing the best, so we used this as a positive control and for further experiments and for the validation experiments. Then we wanted to determine the limit of detection, and then we generated a PTPN12 knockout pool using the CRISPR RNA that I mentioned before, and spiked the knockout cells with wild-type cells, and evaluated when we still obtain significant results. And here for the soft agar, we obtained significant results starting from 0.8% edited cells, which corresponds to 20 cells per well. And here in the GILA, we started to see significant results starting from 3.1% edited cells, which corresponds to 79 cells per well. So here we could see that the soft agar is more sensitive than the GILA. And then we wanted to know, how does this correspond to a comparable in vivo experiment? So we did the same here. We spiked again the PTPN12 knockout cells with wild-type cells in different ratios and injected them either subcutaneously or in the mammary fat pad of NSG mice. And we observed them for 5.5 months, and we didn't find any tumor formation. And then we were wondering, what does this mean now? Are the GILA and the soft agar oversensitive? But then we looked at the literature and we found that PTPN12 is one of the main driver mutations in triple-negative breast cancer. It is involved in tumorigenesis of hepatocellular carcinoma, and it's a marker of poor prognosis in human cancers. So you could say that PTPN12 deletion is a significant safety risk. And considering that the engraftment rate of human tumors in NSG mice is highly variable, in this case, the soft agar and the GILA could provide relevant safety information that would have been otherwise missed. And probably in this case, the in vivo study might have been undersensitive. So in conclusion, we established a suitable in vitro system to detect the tumorigenic potential of adherent genome-edited cells. And our system offers many advantages over common in vivo studies. For example, the soft agar takes 1 month, whereas the in vivo study takes 6 months. The soft agar and the GILA are also roughly 30 times cheaper than an in vivo study, and they are universal, so they can be performed with common lab equipment. And they're also 3R compliant. Regarding potential applications, so we are using it to test the safety of ex vivo edited adherent cells. However, I cannot go into specific applications here. And since the MCF-DNA are already expressing the Cas9 and we just need to electroporate, the system is super easy, so it can be used as a screening tool. And currently we are also investigating other cell lines, and we want to know how translatable are the results that we have in MCF-DNA to other cell lines. And ultimately, if MCF-DNA can even be used as surrogate cell line to test cells in suspension. We are also currently preparing a publication because we like to spread the knowledge of this new method to gain acceptance. Because also in theory, the health authorities are very open to new in vitro system, particularly for cell and gene therapy. But before this can be a regulatory assay, we need to do way more validation. For example, we need to do cross-lab validation, which we initiated. And last but not least, I would like to mention that we also collaborate a lot. So at this point, I would acknowledge everyone. And I would like to thank the organizers for giving me the opportunity to present my data. And now I'm happy to take any questions.